Thanks so much. I should preface all this by saying that I'm the least medical expert person that you're probably going to hear from today. The good news is I won't use any long words, or at least not nearly as long as the ones you've heard already. The bad news is I probably don't have as many answers. Before I get in to a little bit more detail on the Rare Cancer Research Foundation and the work that we're doing there, I think I want to take a moment to tell a little bit of my own story, because this particular community is one that's pretty close to my heart. It was 2012, and I was living in China at the time, and actually in a business meeting in Beijing, when I pretty abruptly lost the vision in my right eye. And made my way through the meeting, figured that I probably had something in my eye, would embarrass myself. And if any of you have ever been to Beijing before, you'll know that most of the office towers have shopping malls at the bottom. So I went down to the shopping mall, walked into a lens crafters, and said, I can't see out of my right eye anymore. Can you check and see if something's wrong? The guy peered in for a minute and said, no, I don't see anything. Maybe try reading glasses. I said I didn't think that was quite the right problem. So 5 doctors and 48 hours later, I was sitting with the dean of ophthalmology at one of China's big medical schools. And she said, I think you might have an apigmented uveal melanoma, but I've never seen one before, so I'm not sure. To which I responded, I think it's time for me to go home. So I basically Googled ocular melanoma in a cab in Beijing on the way to the airport, and sent out a message to a group of friends saying, the most published people I can find on this disease are these people called the Shields at this place called Will's Eye in Philadelphia. I'm headed to the airport now. By the time I land in 24 hours, I need someone to have gotten me an appointment. And I ultimately went through treatment and am a little bit more than 3.5 years out now, and am doing well. But along that path, as I learned more and more about ocular melanoma, and compared and contrasted it to my father's experience battling brain cancer about 3 years before I was diagnosed, and compared that to another good friend's experience battling a brainstem bone cancer called chordoma a few years before that, I was struck by the fact that the stories were all really similar. You had diseases that hadn't necessarily made as much progress, or the research communities hadn't necessarily made as much progress as some of the other more common cancers. And a lot of the problems that were articulated weren't unique to the disease. It wasn't that there was this really novel, unlike anything else, scientific problem that no one had heard of before. Instead, it was, I can't get access to tissue samples. I can't get access to cell lines to do my tests. I can't get access to animal models to do tests. I can't do genome sequencing to learn more about this disease. And those problems aren't unique to ocular melanoma or to brain cancer or to lung cancer or to breast cancer. Instead, in many ways, I think they're most easily thought of as infrastructural problems or utility problems. Essentially, you need certain things in cancer research to be the equivalent of making sure the lights are on and the water's running. And so we decided, as we sort of asked those questions again and again, we recognized that there were places where different disease-specific foundations, like the Ocular Melanoma Foundation, were essentially being forced to reinvent the wheel again and again, because that infrastructure didn't exist for their disease, and so they were having to build a whole they had to build the power plant and the water treatment facility and all these other things before they could even start to do the interesting work. And so that's when we decided to create the Rare Cancer Research Foundation. And essentially what we do is we try and build services or solutions that all of these different disease-specific foundations like OMF can use to do their work more efficiently and effectively and quickly, so that hopefully we can bring new treatment innovations to patient communities, to all of us, more quickly than they would have gotten to us otherwise. We started off by doing that with cell lines. So if you imagine for a moment that you're a researcher and you've got an idea about a treatment that you think might work for ocular melanoma. The first thing you might want to try and do is see whether there's any genetic or genomic information that's already published that would say, yes, there's a good match. The next thing you might want to do is test essentially that treatment in a petri dish. So simplifying a little bit, imagine that you need some cells that you can that you can basically apply your drug or your treatment to and see if it works or not. It can be really hard to get access to those types of cell lines. And so over the last few months, we've been working with the Ocular Melanoma Foundation to get a number of these cell lines deposited from the Curie Institute in Paris with an organization called ATCC, which is basically a very big nonprofit that basically will send research materials to anyone who wants them. But by basically working to get these cell lines out of this one laboratory in France and into this distribution partner, we can make all the other researchers in this room or outside of this room more able to quickly get access to cells that they can use to test drugs that may be beneficial. Let's say then that the outcome's pretty good. The next solution you may want to try and do is test that same treatment in an animal model. So imagine that you have a mouse or a rat or a dog, or there's any other number of possibilities that you want to try and test your treatment in as we try and figure out whether it might work well. We work with Jackson Laboratories and Charles River and some other organizations that specialize in creating those types of models to create publicly available models that researchers can use to try and test new treatments more quickly and cost effectively. And let's say then we get more good news and we're ready to try it in people. We're working right now to try and make it easier to essentially help patients looking for clinical trials to identify which clinical trials they're eligible for and can enroll in. Clinicaltrials.gov, as we talked about in the last session, has tons and tons of information in it, but it can also be pretty hard to make your way through it. And there's certain organizations that have already done some really neat innovations around making it easier and quicker and more efficient to match patients to really great clinical trial opportunities. The Michael J. Fox Foundation, for example, has been doing this in specific things not related to cancer, but the problem is still the same. You have some set of attributes of yourself and your disease, and different drugs and treatments are basically good at targeting different attributes. And if you can do that matchmaking process more efficiently, it means that both the researchers who have new treatments and the patients who are looking for the best treatments can find each other more effectively. So long story short, we've been excited to try and bring some of those different types of innovations on helping patients to donate tissue to research, to helping researchers get access to all these different things like cell lines and animal models, to even helping patients and doctors find each other when a doctor's treatment looks like it could be a good option for a given patient. And because we're doing it in sort of a infrastructure way, it means that we can do it for a whole bunch of different cancers at the same time. One of the things that we focused on as we thought about these solutions is that even as this ocular melanoma community can feel small, rare cancers as a group represent the leading cause of cancer death in the United States. Together, we're a really big number. There's 400,000 or so people diagnosed in the United States every year with a rare cancer. And if we can build solutions that work and are flexible to be helpful with ocular melanoma and chordoma and glioblastoma, and the list goes on, then what we can do is actually attract resources from cancer centers, from large philanthropic donors, from pharmaceutical companies, that it's been hard for ocular melanoma to attract by itself. But if we can basically make it so that they're able to go after that larger population, the 700 or so different rare cancers on the list, all at the same time, or at least a larger portion of them, lo and behold, we can attract resources and attention to working on rarer cancers like ocular melanoma that haven't gotten so much attention from those same organizations before. As we look forward, there's a few more things that we're really focused on right now. We've recruited a whole Silicon Valley style technology team to try and make it easier for patients to agree online to donate some combination of a tissue sample or their tumor sample after a surgery and medical records to researchers so that we can more quickly do new development of new therapies, either because we can use it to do better genome testing or we can create cell lines and mouse models, or we can even just use that to help more quickly match the patient based on their medical records and on their tissue sample to the treatments or combinations of drugs that could be most promising for them. And we're excited to potentially be doing more work with this community in that area. In parallel, we're also excited to build on the first 6 cell lines that we got from the Curie Institute are going to be publicly available by the spring. We've got 3 more that we're trying to get from the Netherlands right now, and then there's others we're hoping we'll be able to get over time, all with this goal of getting more research materials available and accessible so that the doctors and the researchers who are trying to develop the next generation of treatments for us can do that work more quickly and cost effectively. Because in the process, what we can do is get a lot more people to work on this disease or this type of disease than have been doing it in the past. Right now, if you imagine yourself being that young researcher, if you see a disease that doesn't have cell lines, that doesn't have animal models, that may not have genomic information, where it's hard to find patients and match them to diseases, lo and behold, they say, I think I'll go work on breast cancer, thank you very much. But if you can find a way to make that effort easier, say, sure, here's cell lines, here's the animal models you need, we're ready to let you do the interesting work, not the work of trying to reinvent the wheel. You get to build on top of that foundation. It's much easier to attract research attention to the disease and hopefully make more progress more quickly. I could go on a long time about all these different things, but I'm also sensitive that I'm probably going to be more fun and interesting if I answer your questions than if I talk about the way I'm spending my day these days. Are the things I can talk about either from the perspective of having gone through my own patient trajectory over the last few years, or in particular, the ways we've been trying to work through the Rare Cancer Research Foundation to accelerate the process of going from an initial hypothesis about a potential treatment all the way through to testing that treatment and hopefully extending patients' lives. Yes?
What kind of tissue do you need to collect from a patient to put into that storage cell or whatnot? Does it need to be blood, vitreous, or?
So the answer to that varies by cancer is the first answer. As a general rule, what we want to try and do is get a blood sample that is essentially a healthy tissue baseline and then a tumor sample. Obviously, in the case of folks that don't have metastatic disease but have ocular melanoma, it may be difficult to get a tumor sample in a traditional sense from the eye unless you end up with enucleation, which hopefully we can avoid more and more. So in that sense, there's a challenge right now in ocular melanoma specifically about how do you get a tissue sample for a tumor if you use radiation and you don't actually have surgical removal. Right now, the answer would be we can do it more easily in metastatic disease. We can end up with a liver met, a healthy blood sample, and then your medical record that says this is the treatment you got, and this is the treatment you're getting, and your outcome. And we put those things together, which, believe it or not, is really, really hard to do. Then you end up being able to get a lot more insight into essentially, if you imagine, if I have a profile of you as a patient, of your tumor, and of your treatments and outcomes, when I can put those pieces of the puzzle together, you can get much more smart, much more quickly than if you have to do tests on just one of those things, or have to try and understand a disease or a treatment based on just one or two of those things. I don't know if that made sense, but broadly at least. Other questions? I've made the room go silent. Uh-oh. Yes.
In that point, I mean, this is what's needed for many of things, and you're hitting the right ones. Fantastic.
Thank you. Thank you.
Yeah, out of your personal experience, so can I ask a question?
Of course.
Did you get your sight back? And if so, did they identify what caused the loss of sight?
Yeah, so in my specific case, somewhat ironically, I had exceptionally good vision before this. I literally had 20/10 vision, which meant that I didn't regularly see an eye doctor. And so what had happened was over time, my tumor had essentially increased pressure on my retina, and eventually my retina detached, which is what caused me to pretty abruptly lose a good deal more vision. When my retina healed, I got a little bit of vision back, call it like, a third of my total vision. And so the curve was essentially lost almost all of it from the trauma, then it reattached and I got a little bit back, but then I've also had some degradation of the vision that I had left ever since because of the radiation damage. So I'm fortunate that my left eye still treats me pretty well, but my right eye isn't helping out so much at this point.
Is there a contact information or something that we could get? I had my eye removed. I had a nucleation 7.5 years ago.
Yep.
And my tissue blood samples are still at the pathologist's
Yep.
lab. You know, I mean, is there a way that I could donate those?
Absolutely. So there's a
My
couple.
medical record or whatever. I wouldn't care to do that.
Perfect. Well, that's super helpful. The answer right now is that we aren't live with OMF on the platform that the Rare Cancer Research Foundation is running. So there may be a broader question that I should connect with Grant about or others in this room to think about how we can try and do that effectively. The biggest thing that we have focused on is especially on fresh tissue. That can still be turned into cell lines and animal models. The stored tissue that's been biobanked somewhere can be really valuable as well, especially on sort of genetic and genomic pieces. Right now, our mission at some level is to do remember, we're sort of the utility here, which means the way we get into we'll call it the business of collecting a specific cancer's tissue is when a foundation or a research lab or a cancer center or someone says, I really need more tissue to do my work, and then we go out and find that tissue for them using a combination of sort of an online, called the DocuSign for Medicine, an online consent platform, and then agreements with a bunch of different cancer centers to try and aggregate tissue. Coincidentally, we're at rcrf.org, so we're pretty easy to track down from that perspective. Other questions that I can answer, personally or about the foundation? I think that probably then is my time. One thing I'll say in closing, a not particularly eloquent researcher in February of 2012 when I first got diagnosed, when I was emailing around trying to find anyone I could find with expertise, said, I said, what treatment should I get? I've got a pretty thick tumor, my prognosis isn't great. And he said, quote, I don't have anything for you now, but there's some great stuff coming in a couple years. That's not the best thing to say to a new patient. I could tell he was the laboratory researcher, not the clinician. But one of the really fun and exciting things, I think, is that it now is 2 years later. It now is, in fact, for me, 3.5 years since my initial diagnosis. And the transformation going on in oncology medicine generally. You saw references to targeted therapies, to immunotherapies that is happening right now mean that the conversations that I'm the layperson, not the doctor, but the conversations I'm listening to doctors have today, even compared to those had only a few years ago, are remarkably more promising than the ones that we're having not so long ago. And especially as we start to get more of this infrastructure that lets us do that work, or lets, frankly, not us, but the researchers that are way smarter than me, do that work even more quickly than ever before, I think offers a really, really, really powerful chance that if, I actually had my last MRI last week, and originally they said they thought saw something, and then they changed their mind and said they didn't, which is always a fun process. The probability that there's something that can come to market, come to us as patients in time to affect my outcome, I think is much, much more probable now than it was only a few years ago. And I'm really excited by the work that OMF is doing, that we're able to support in certain ways to try and make that process of bringing good ideas to patient communities more quickly a reality, because I think we're seeing the needle move faster than we have in a really, really long time in this disease. There's a range of bad ways to say it, but if one were to have to become an ocular melanoma patient, now's the best time compared to any time in the past. And I guess I just would close in saying that I'm really excited about and inspired by the work that researchers that are here today and that the foundation are doing to try and help us bring those research innovations to our clinics and to our treatment protocols more quickly, because I think it gives all of us, myself included, a much better chance of battling this disease successfully. Thank you all for having me today.